Thanks, Ruth. Thanks, everybody, for coming out tonight. It's really good to see a full house, and thanks to our speakers for taking time to present their stories tonight. It's really a pleasure to introduce tonight's speakers for a couple of reasons. So first, you saw the slideshow here earlier. They're recipients of the Sequoia Park Zoo Conservation Grant. So they worked hard and applied for different types of funding. Some came from our zoo to support the work that they're going to share tonight. So we're especially excited to hear about the work that they've done because the zoo feels like we've played a small role in helping it be successful. I also am happy to introduce them as colleagues of mine at Cal Poly Humboldt. And not just colleagues, but representing really a wonderful symbol of collaboration. So we've got Dr. David Sinn, who's a colleague of mine in the Department of Wildlife. He's been teaching at Cal Poly Humboldt for about 3 years. And he comes with a marine biology undergraduate degree, but really matured in his sciences by studying everything from small little octopus and shrimp up to the exciting Tasmanian devil. So he's really worked on the behavioral ecology of vertebrates and invertebrates, and is really a broad ecologist, well-trained in all areas, and kind of getting back to his marine biology roots. And then he's collaborated with a faculty member in fisheries, Dr. Rafael Cuevas Uribe, who has been working at Cal Poly Humboldt for over 10 years. And he's been also has worked on a variety of fish species, really specializing in aquaculture. So how do we raise fish? And so his expertise came into this Seastar story as how we can propagate these species. And finally, we have Dr. Sean Craig, I was going to say one of my oldest colleagues, longest standing colleagues. We've been working together for the 20 years that I've been at Humboldt in the Department of Biology, zoology as a marine biologist. And Dr. Craig has worked on invertebrates and also eelgrass, working here in Humboldt Bay and off the coast in, again, a variety with decades of research on a variety of marine issues. So it's really such a great collaboration between these partners, and I'm really excited to hear about it. Oh, and they included students in the story, which I think is really special, and some of them are here in the audience. So thanks for participating and coming. And without further ado, I'll turn it over to David. Thanks.
Thank you, Michaela. It's very kind. And I want to just thank you in general for organizing the speaker series. I love the connections that we can have with our community here. And thanks to Dr. Ruth Mock for her work with the Zoo's Conservation Fund and the Conservation Council. I'm going to be looking back at the screen tonight, looking at my notes because I'm very nervous. So I'll try not to do that too much, but bear with me. Our story tonight is actually about kelp, bull kelp and giant kelp, to be more specific. And these two kelp species are actually the largest brown algae in the world. Giant kelp grow up to 45 meters in length in some cases, and bull kelp are not that far behind them. They also grow into the tens of meters. Some of these species can grow up to 2 feet a day. So they are a really remarkable marine organism. Biologically and physically, these guys are responsible for what humans call a kelp forest, which are some of the most biologically diverse and productive nearshore reef ecosystems right across the globe. So they're really important not only for marine biodiversity, but if you think about all of the organisms that depend on kelp for their livelihoods and the humans that in turn depend on those marine organisms, they're also important for human cultures. The larger story of kelp across the world, and especially here in Northern California across the past few decades, is not a very happy one. Kelp populations have been decimated in recent history, and oftentimes multiple stressors have turned our biologically diverse, productive kelp ecosystems into some other type of ecosystem state. Oftentimes, what you'll see are a lot of purple sea urchins grazing on some barren rocks. Purple sea urchins are one of the dominant herbivores that feed on giant kelp. And then there is the star of our story tonight, the California sunflower Seastar. It turns out that sunflower stars are part of the story of the multiple stressors for how we're going from these diverse productive ecosystems into something that objectively is less diverse and less productive, and maybe from our standpoint, arguably less valuable. The sunflower stars are the main subject of the work that we got for funding from the Sequoia Park Zoo, and it's going to be the main subject of our talk tonight. But before we get there, I want to take a few moments and acknowledge the people and institutions that were critical for this work. Sequoia Park Zoo Conservation Fund for the seed grant that they gave us to get started on this work. Telonicher Marine Lab for their in-kind support, facilities, seawater, help with maintenance. We couldn't have done it without these two institutions. I want to mention a nonprofit organization. It's called the Sunflower Star Lab. Their director, Ashley Kidd, they actually are a really important part of the larger North American story of sunflower star conservation. They are dedicated to organizing tens of institutions and researchers to make a concerted effort for sunflower star conservation. And they were really critical for us because not only did they welcome us with kind of open arms, but they also put us in touch with many researchers and expertise that was already occurring across North America. And so a few of those that have really played a role in our work are Friday Harbor Lab, Moss Landing Marine Lab, and the California Academy of Sciences. And then I just want to mention a few folks up at Telonicher Marine Lab, Grant Everly, Kyle Weiss, and Allison Formica, who have been really important helping us on the ground and helping us to construct what Rafael will talk to you about later, our marine invertebrate larval rearing system. Now, our title has students in the title, and that was very intentional. Part of why we set out to do this work was we felt like there was an opportunity to combine conservation with important training opportunities for students. And a few of these folks I want to mention in particular, Colby Pitt and Cody Carlson. These two were undergraduates who were there at the beginning of our work, who helped initiate, brainstorm, run experiments, essential to the things that we're presenting to you tonight. And they even took the initiative and went beyond and did extra stuff, like starting a sunflower star sighting network and organizing other volunteers from Cal Poly to get involved in this work. I also want to mention Kai McClave, who's been really seminal more recently in helping us with holding some juvenile sea stars up at Telonicher. And I just want to say that all of these students had an opportunity to learn something about marine aquaculture, in this case, echinoderm aquaculture. So I already mentioned to you the larger story. We've got giant kelp here in the bottom left and bull kelp up there in the top right. These two species co-occur across the northeastern Pacific, all the way up to the Aleutian Islands and down into Mexico. Giant kelp actually has a more global distribution. Anywhere in temperate or subpolar latitudes where the conditions are correct, you can find giant kelp. there's a couple of things that I want to talk to you about kelp that is going to set the stage for tonight. And it's about their structure. They have these little balls along the stem here, and you can see the big ball there on the bull kelp. These are something called a pneumatocyst. It's essentially a hollow ball that the kelp fills with air that allows the fronds to float. Why is that important? Well, if you start to create a bunch of 3D structure in the water column, all of a sudden you create spaces where fish can hide, hunt, mate, do the things that fish like to do. The other important structural component of kelp are what's called a holdfast. And these are where the algae attaches to the rocky substrates. And this in turn provides really good habitat for the marine invertebrates that are really important for the fish, that are important for our human communities. So I mentioned that the contemporary story about kelp is not a very happy one. In places like Northern California, Chile, and Tasmania, kelp species have shown severe declines in the recent decades. And what we know about these declines is that it has something to do with this idea of multiple stressors. There's a story about water temperature and how water temperature is related to nutrient availability. There's also a story about grazing, and that's going to take us into our work tonight. And so let me just touch base with you on the idea of sea surface temperatures. So this is a graph of the sea surface temperature along the California coastline. And what this does is this graphs the average sea surface temperature at a given site over a year, and it compares that to a running 35-year average. And what you can see here is that in July of 2014, our waters experienced a pretty massive heat wave. So from the period of July 2014 up until June of 2016, this is the most recent marine heat wave that is thought to have really seriously affected our kelp. And in fact, in October of 2015, that is the highest sea surface temperature average that has been observed over the last 35 years. Well, okay, 2 to 4 degrees Celsius. I suspect most of you are educated. You know that matters, but it seems like not too much, right? But to a kelp, this means this makes a big difference. And so if we look at kelp canopy coverage, so this is the amount of kelp that's actually grown to the surface, and that we can measure the amount of surface area it's taken up. You can see here, back in 2013 or so, we had almost 2,000 square kilometers of canopy kelp coverage from Jenna up to north of Fort Bragg. And I'm just using this section of coastline because it's been well characterized, but this is the pattern up and down most of the California coast. You can see that the kelp canopy coverage beginning sometime in 2014, when that heat wave hit, started to decline all the way down here to 2019, when we had 2 square kilometers of kelp canopy along this pretty significant stretch of coast. And if you're like me, you like pictures instead of graphs. Well, I like graphs too, but maybe you like pictures. These are actually satellite pictures taken by NASA, and you can see kelp canopy forests from the sky, okay? And this is a picture taken of that same area in 2008, and once again in 2019. And to put a little bit of an emphasis on that, someone came in with a Sharpie and colored in all those light green areas for us. But you can see without a doubt that kelp along the California coastlines are in trouble, okay? So I mentioned warming waters, and I mentioned nutrient availability. Well, oceans go through a lot of variation in their sea surface temperatures. Kelp numbers actually go up and down. So why have our kelp not actually recovered from this marine heat wave that we experienced back in 2014? And I just want to point out to you that population disappearance or extirpation and species extinction, what we know is that it rarely occurs from a single stressor. Okay, usually what happens is that there's something called synergistic effects. This is when multiple stressors interact, and that interactive effect is greater than the sum of the individual parts. Okay, so we're going to be talking about sunflower stars tonight. They're one of the synergistic effects that are resulting in the lack of kelp that we currently see along our coastlines. So we've got sea surface temperatures. Those are tightly bound up with nutrient availability, so I'm not going to try to break that down. What I do want to talk about is grazing, because the other thing that's gone on is that we've witnessed an explosion in terms of numbers of our purple sea urchins along the California coast. And these guys are the dominant herbivores that eat kelp. Their story is kind of interesting too. Prior to the 1700s, one of their major predators was the sea otters. Okay, and we know the story about sea otters. Unfortunately, they have yet to recover. And so we're not going to talk about them tonight, even though we still love them. Since sea otters have gone away, we think that the organism that has been largely responsible for keeping sea urchin numbers in check is the star of tonight's show, the sunflower sea star, Pycnopodia helianthoides. Okay, these guys are the ones that we think have been a part of the regulation of the sea urchins that have been eating kelp. Okay, the problem is the sea stars have gone away as well. And that's also going to be part of our story tonight, the main story. And so this is where the three of us started in 2022, coming together, meeting one another, talking about surf spots in Mexico, and this led to talking about conservation in our area and the basic decimation of what we're seeing across our coastlines as these kelp forests get transformed into something else that's either rocky and barren or covered in purple sea urchins. And that's where I think I'll hand it off to Sean.
All right. Great, thank you.
Yeah.
Okay. It's my pleasure to go next. It's been a great pleasure to work with these two gentlemen, and I'm going to take a slight step back here to start by saying, what is an echinoderm? We've been talking about echinoderms. What the heck is one? Well, short answer, they're very strange animals, to be honest, especially here amongst this audience at the zoo. But let me explain. They're basically animals with spiny skin. And some of those animals are obvious. The sea urchins obviously have spiny skin. Here's a close-up of their skin. And I'm actually going to pass around a red urchin that still has its spines in place. So you can see that. I ask that you very gently carry it. Try not to drop it. This is our one specimen that's still whole. And so they've got a spiny skin, all of these animals. The sea stars, believe it or not, have a spiny skin. The brittle stars have a spiny skin. Even these guys, which are essentially flat sea urchins, have a spiny skin. I brought a little trick here to convince you of this. If you take a look carefully under a microscope at the surface of these sand dollars, they're actually just flat urchins that have a 5 o'clock shadow. And so that's the spines. You just can't see them as well. And I haven't put up on the slide here sea cucumbers or sea lilies, but they also have spines. So spiny animals, and they also have generally pentaradial symmetry, meaning they're radially laid out, and usually there are 5 arms, although we're going to break that rule in just a minute. And in addition, they actually move with hydraulic power. They will pump water into their bodies, and that comes in here, in this structure right here called mammalia. The water comes in through there, down a canal, around the mouth, and then out into each arm through what we call a radial canal. And then it's pumped out through a lateral canal into these bulbs here, which are kind of like the bulb on the end of your turkey baster. And the muscles in here contract to pump water into the tube foot, and that's what allows them to stretch out a tube foot. And then each tube foot will contract a muscle here to suck on, and literally one by one, they pull themselves along, usually pretty darn slowly. And then last but not least, these animals are made up of calcareous plates. Their skeleton, internal skeleton or endoskeleton, is made of plates. And believe it or not, when those plates are all connected, fused together in a sea urchin, we get something like this. This is called a test. I'm going to pass that around. It's even more fragile, so please be careful. I promise I won't give you all a test on the test, but my students are literally getting one tomorrow. And that's basically the kind of that's a short version. And one of the important things about echinoderms and most marine animals, especially the invertebrates, as well as the invertebrates, is that they have a complex life cycle. They have a part of their life cycle that really is spent a great deal right here up in the water column. And it's a microscopic stage. It's a series of stages, really. So a typical Seastar will be either male or female, releasing sperm or eggs. Fertilization will lead to developing zygote that then goes through a few stages here, starts to develop a gut, and then completes that development as an early larva. So that the larval phase here literally has a mouth and a digestive tract and an anus, a full digestive tract. Why? Because they're feeding in the plankton. They have cilia on them that are directing phytoplankton into their mouths. They are eating while they're up in the water column. And if they get enough food, then they will continue to develop here, and they essentially develop longer arms, which are coated with more cilia, so they can beat those cilia even more and create more currents and feed more. And ultimately, then they will settle to the bottom and drop off this little guy that we know of as a little baby Seastar. And that's their development. And notice that if we want to know something about their populations, ideally, we really want to know what's happening in the water column and not just the number of adults, right? Okay. But why do we care about Seastars? Well, we care about Seastars because many of them are what we call keystone species. And a keystone is the one stone in an arch, which you can play the game of Jenga with and lose, because you're going to slide that one stone out and the whole arch will collapse. That's the idea of a keystone. So a keystone species is a species that might not be that abundant, but whose presence and activity is really, really important, okay? And we know this from some research by this gentleman here, Bob Payne, a professor at University of Washington, who did a very, very long series of experiments here, literally 10 years long, where in some areas on the coast, he removed, as you can see here, all of the Seastars. In this case, they were this overstar. And in areas where he removed the Seastars, areas without Piesaster generally dropped dramatically in a number of species. So the communities drop, they sort of collapse down into very few species. Whereas if we leave the Piesaster there feeding, we get many more species. So much richer, diverse community with Seastars present. And this is the same story here visually. And what I simply want to show you is that in this case here, the Seastar clytaster is feeding on these mussels. And if it doesn't feed the mussels, if we remove them here, then we get nothing but mussels coating the shore. And that's why the species richness drops. We've got literally one species left. So ultimately, we have keystone species, again, that one species that's more important than the others. If we remove it, the system collapses down to relatively few species. And that, of course, where our star of the show today comes in, the sunflower star, right? Pycnopodia helianthoides, which, first of all, breaks the rule, right? It has 23 arms, not 5 or multiples of 5. It is a very rapid moving sea star. It is a very large sea star. I've seen sea stars a meter or more across underwater. Okay, and they're very, very common. Well, they used to be. Okay, and what happened to them? Well, essentially, they got the Wicked Witch of the West treatment. They melted down in place. They gained a disease called Seastar wasting disease. And what Seastar wasting disease does is literally sort of melt them down in place. This is an actual picture of a sea star, and that's, well, what's left of it here after it's been hit by Seastar wasting disease. And Seastar wasting disease starts out as a small lesion in the upper surface. The skin is actually coating the skeleton. So the skin of that urchin that I passed around would normally be coating all of what you felt. And that begins to form lesions. It begins to slough off, and then eventually the whole animal starts twisting its arms, and ultimately the whole thing melts down in place, and we get some sort of whitish goo left behind on the rocks, literally. Now, this Seastar wasting disease hit a lot of sea star species, not just this one sunflower star. And so you can see here listed a number of different species. Most of these species have recovered, but unfortunately, the sunflower star has not. And what we know about it today is different from just a few years back. What we know today is that there's a particular species of bacteria, Vibrio pectinacida, that causes Seastar wasting disease. We can take that bacteria, we can take a syringe and draw out the fluid from a sea star, and we can boil it and kill the bacteria, and no problem injecting it into a number of sea stars, they're fine. But if we don't boil it, they will all gain this Seastar wasting disease. And the Seastar wasting disease has been absolutely devastating. If we plot the changes in the populations of these sunflower stars all along the entire Pacific coast, we see drops from 100%, 90%, 99%, 100%, 99% here in our area. Essentially, all the sea stars have been wiped out. And I say that to you today confidently. Why? Because I have a team of divers that works with me. They go out every summer, all summer long, the days that they can dive, of course, and they're out there counting sea stars and other things. And the numbers of sea stars we've seen in the last 10 years can be reflected on the fingers of one hand, about 3. Okay. So they're gone. They're effectively gone. And that leads to the not so comical little cartoon here. Hey, wow, this party is really heating up. Heating again. This is a coincidence, right? The marine heat wave that David talked about. And what happens now? Well, the sunflower stars are gone. The others are left wondering where they went. Right. At least in the cartoon. Okay. And I'm going to pass it off now to Rafael.
All right, thank you. Okay, so what is the goal of this project? What did we do with the ponds? So basically, we wanted to build a echinoderm system to demonstrate why it's important to protect this endangered sunflower sea star. And we want the students to learn to raise them in captivity. We want them to get experience of building a system. We want also, the students to learn not just rearing sunflower Seastars, but other echinoderms too. And at the end of the day, we are all about hands-on learning and training. We want those students to become a help for the ocean conservation. So the system consists of 3 8-gallon tanks. Each one of them holds 8 of 8-inch incubators or nursery tanks. We, the students, plumb everything. So they were able to finish that system in January of 2024. Here is another pictures. Once full of the tanks, now you can see all those 8 incubators. The incubators are called upwellers because the water is flowing from below and then it's coming out from the top. On the back of the system, you can recognize that we have different cartilage filters that is filtered down the seawater from 10 to 5 all the way to 1 micron. And also we are going to have a UV system. So the UV system is going to allow to make sure that the system is stereo. Now, the first organism that the student wanted to spawn was the sand otters. And they successfully spawned them, and they were keeping track of the larvae as they are developing. This was the first organism, and there was some problems with the system, some overflowing. Some of the screens was a little bit too big. The sad thing is none of these larvae survived. Now, during that time, in the spring of 2024, I was teaching my advanced aquaculture class. And here we have Colby, which is in the audience, and Colby, they decided to do a project for that class. Their project was to spawn the purple sea urchins, the red sea urchins, and the bat stars. And you can see them injecting these chemicals to allow them to release the eggs and the sperm. And they successfully spawned them and fertilize the eggs with the sperm that they got. You can see the class, how they're watching, very interesting. And they were keeping track as how this larvae was developing throughout time. At the same time, they were worried about, we need to start growing the algae that they're going to need to eat. So they ended up learning, how to grow different microalgae, for example, Dunaliena and Rhodomonas. And you can see here some of the algae that they were culturing. And as you can see here, what happened is, at the beginning, they have tons of larvae. As the time progressed, it was less and less and less. And probably, 10 days later, almost nothing left. But again, we are about the learning. And what happened next is let's find something else to spawn. So they got some sea cucumbers, they used to spawn them, but the same story. After a few days, that larvae did not survive. But something happened during Valentine's Day of 2024. We had the Technopolia, or the Sunflower Recovery Working Group, that consists of different institutions, and they met at the Birch Aquarium, and they were able to successfully spawn adult sunflower Seastars together with the Aquarium of the Pacific, the California Academy of Science, the San Diego Zoo, and of course the Sunflower Star Labs. So they created, and they call it this Cupid cohort. So they got thousands of larvae. And what happened is, I was teaching in the fall of 2024, my marine aquaculture, mariculture class. And I take my students for a field trip all the way to Moss Landing and to the Monterey Aquarium. So while we were touring Moss Landing, Andrew King chose this little jar over here, this little flask. And he's like, oh, I have a lot of Pictopodia or sunflower Seastar larvae here. And I'm looking into this and I'm like, that's no way, I say that, oh, I have not feeding them, so that's why they are not growing. They are kind of arrested at the larvae stage. I'm like, okay, well, if that is true, let's get some to Humboldt. So what happened is we had to ask, the fish and wildlife, and they just told us to fill out a chain of custody form. And during the California Seaweed Festival that happened last year, Andrew and Ashley, they drove all the way from Moss Landing, and we got 6 juveniles of sunflower Seastar. And of course, the students were very excited, I included myself. I mean, it's like, wow. And they started naming them right away. It has been 10 years since the Marine Lab did not have any sunflower Seastars. The last ones ended up dying in 2014. So after 10 years, these were the first individuals that came back to the Marine Lab. So right away, some students decided, I want to work with them, let's do a project. So we have a new group of students for my mariculture class, and they decided to compare 2 different diets and see how well does they grow if you just feed them sea monkeys, known as Artemia, or if you feed them brine shrimp. I don't know if you can see the little frozen brine shrimp. And they wanted to monitor their length as they were growing. At the end of the day, you can feed them sea monkeys or brine shrimp. There was no difference in growth. They like to eat whatever you toss at them. And after that, they decided just to switch into feeding them frozen squid. And that's the diet that most of the time they are feeding the squid. Now, going back into other echinoderms, it's like, well, let's try to spawn one more time those sea urchins. And you are wondering, like, why do you do that? You just told us that there is thousands of them in the ocean. Why don't you just go there and collect them? Well, remember, whenever you bring a specimen from outside from the wild, they could be exposed already to Vibrio or other pathogens. And, we don't want to introduce anything from the outside into our system that has, clean water. And also, we want to start training those Pinopodias to start eating those little sea urchins as they are growing. So that's the purpose of trying to produce our own sea urchins at the Marine Lab. Unfortunately, this time, just the females are the ones who release the eggs, and none of the males release the sperm, so they could not get any larvae from this experiment. Now, oops. Let's see. Okay. What happened is this semester, because we just have 6 individuals, it's kind of hard to collect like real data, that make it a statistical difference. So we were asking like, sunflower Seastars, we want more. So the California Academy of Science say like, oh, we still have some of those cohorts, the Cupid cohorts. You want some? Yes, how many can you give us? Well, we're going to give you 15. So this is a class we drove all the way to San Francisco to the California Academy of Science, and we got 15 sunflower Seastars. Now, the project that the students this semester they were doing is we want to compare how well does they grow if they don't have any light. That's why you see this tarp over here, so they can cover them. Or if we expose them to 12 hours of light and 12 hours of dark, or just having with light. So that was an experiment. This is the first time somebody's experimenting with photoperiod. And unfortunately, the ones that were with no light ended up becoming stressed, and some of them were start to lose their arms. And some of them also showed symptoms such as wasting symptoms. So right away, we decided like, hey, let's just stop the experiment. I think that's stressful. We want to keep those Pinopodias alive. Unfortunately, these two did not make it. We lost them. But we also were trying to figure out if this was Vibrio that causes this mortality. So we brought them to the lab and we were trying to isolate the bacteria. Unfortunately, we did not have any results. It was inconclusive if this was Vibrio or not. But again, it's a learning experience that the students get from reading them, taking care of them, and also even trying to diagnose the disease. At the end of the day, the story starts because kelp, kelp is the one that has been impactful. My research for the past 5 years has been growing kelp in Humboldt Bay, including ball kelp. And we are here in Humboldt Bay. We are here where we have a farm. The university has a farm to grow seaweed. And this is the best place to do this type of research. And I think Sean is coming next.
And I mentioned earlier that our team has been underwater censusing different areas underwater here along the coast of northern coast of California, all the way up to the Oregon border. And I just want to plant a seed, a thought in your head, which is where we hope to go eventually. Why not outplant kelp? Why not remove sea urchins, which is what our scuba diver is literally doing all summer long, this summer and next. We have a grant through the California Sea Grant. But last, and certainly not necessarily least, why not get the predators back, right? Why not get them working for us too? So I just want to leave you with that thought. That's where we hope to go eventually. David will finish us off.
Yeah, thank you. Yeah, the end goal in sunflower star conservation is, of course, to get them back into the wild. Who knows what's going to happen in the future? I want to point out and kind of wrap up tonight's talk by just telling people how important the seed grant money that organizations like Sequoia Park Zoo are contributing, right? We now have infrastructure, Telonicher Marine Lab, can be used to raise a wide variety of different types of marine larvae. We now have some sunflower Seastars in the lab at Telonicher. We have training opportunities for lots of different students in a field that arguably is going to be growing, aquaculture, as human populations continue to grow. So we don't have a lot of endpoints right now. This work is ongoing, and it is the intention of groups that are involved in this work eventually get sunflower stars back into the wild. Working out how to spawn and rear their larvae in the lab is going to be a critical part of that, as we're going to be needing to do that to hold individuals in the lab. And that's really what the story is tonight. It's how we've gotten started on some of that work here at Humboldt. Thank you for coming out, listening to our talk.
Thank you so much, David and Rafael and Sean. If folks have questions, do you guys want to come up here and
Yeah, sure.
you can field any questions that you might have?
Questions here. Happy to try and answer.
Is it in California working on the immunology of existing wasting disease?
Well, that's a good question.
I'll
Yeah.
try and have a little, I mean, the short answer is yes. We're really just starting to learn a lot about the immunology of most, I would say, marine invertebrate animals. There's not a lot known. And there's so many questions, right? So it's an excellent question because we need to know how these diseases spread. We need to know why they're increasing size.
Can you repeat the question?
Oh, sure, I'm sorry. The question was, is there anyone studying the immunology of these diseases? Short answer was yes. And the longer answer is really that there's lots of questions and not that many good answers, yes. So in a sense, it's taken quite a while. Actually, the initial studies thought that the waste disease was due to a virus, but recently that's been overturned. So continuing work is going on to try and figure out how these diseases spread in the ocean. I fear that that's a very big and important field in the future because that is really, there's every sign that these diseases are increasing in our ocean, corals and sea stars and what have you. So it's an excellent question, and I wish I could tell you more. Yeah.
Very closely related. The area in British Columbia where appears to be a little refuge, we know, are they less susceptible to the virus, or are they just not encountering that?
We don't, yeah, good question. We don't know that yet, but we do know that some sea stars were not susceptible to this disease. There's one, and I'll tell you the story very briefly. There's one little sea star, literally about this big in our area here, that has 6 arms. It used to be called Leptosteris hexactus. And our team knew we were doing the right thing when we were down on our hands and knees counting sea stars. And literally everybody popped up and said, well, I found 20 of these and nothing else, right? So we don't know yet, but people are working on it, whether they can find the genes responsible. And not only that, we are collecting samples from any sunflower stars we now see and shipping them to one of the groups, one of our networks, scientists who's extracting the genes and looking at the DNA, both before crash and after, looking for those genes that might be responsible for protection from this disease, perhaps. So ongoing work. Yeah.
I'll just say these guys are broadcast spawners as well, which means that the gametes get put up into the water column, get mixed, with ocean currents. This population along our northeast coast, they're reproducing with one another. We don't know any sort of genetic isolation.
Right, so there's a lot of connectedness, and that makes the study a little bit more challenging. Yeah, good point.
Did you say at the beginning that there's also populations in Tasmania and some
Giant kelp,
not the sea stars?
not the sea star, but the giant kelp is also.
But are the sea stars anywhere else then? It's not somewhere else?
The sunflower stars are not anywhere else besides this coast. So the story of the collapsing kelp is unfortunately elsewhere as well, including Tasmania.
And purple sea urchins as
Yeah,
well.
and urchins raise them down. That's a repeated theme.
I was just wondering if you're noticing any kind of background levels of Vibrio or doing plating for it in the marine lab.
It's a great question. Do you want to try?
Yeah, I mean, I think everything is going pretty good at the Marine Lab. We have not noticed any big mortalities. Yeah, so, but I don't think nobody's like really monitoring Vibrio per se.
Yeah, we hope to start sampling for DNA in the water column, to see if we can start to pick that up.
Yeah, I mean, so they make like GCPS plates that are really easy to swap for Vibrio.
Right.
And I've noticed that like you do a standard testing protocol like weekly of just like water sampling and swabbing and incubating. I've noticed that, I mean, it's not Vibrio species specific, but.
Well, yeah, but that kind of testing is exactly what we need to be doing next. So we're actively hunting for further grant funding to add some components to this whole project. Yeah. Yeah.
How can the public help?
You want to try that, or do you want me to try that?
Well, it's a good question. I'd like you to add to
Okay.
my response, which probably isn't maybe a very good one, coming to tonight's talk, talking to your neighbors about it. There is, for good reason, a lot of restrictions around moving sunflower stars around and about putting them back in the water. There's good reason to be conservative about those approaches. But I think, coming to things like this actually creates momentum and creates some sort of, reasons for folks to want to see these things back in our waters. This is, when we started, the reason why we started is because it felt like there was a gap here, right? You have to go down to San Francisco before you reach the next marine station south that's doing anything with these organisms. You have to go to Newport before you hit somebody else in the north. So there's this gap along a major chunk of our coastline where no one's looking out for these guys, no one's talking about them, no one's getting out there and doing it. So I don't know, that was a great answer to your question.
Let me just add, I think that is a very good answer, because I think you all can keep your eyes open and let us scientists know what you're seeing, right? I mean, are you seeing sunflower stars? That's a big, important question that we need all of you to help us answer. Obviously, many pairs of eyes are better than a few. And then the other thing is what's happening with the urchins, where you're seeing large numbers of those. And so that's all actively ongoing work, because the more eyes we can have, the more people we can have out there looking, the better we are able to adapt to this change. And frankly, I'll be a little bit more out of the limb here than David went. The more we hear from you about where our community wants to go, the more that will help to push the science focus, right? David just mentioned that we don't have permits to put out any sunflower stars yet. So there's a lot of wishful thinking happening inside of me about what I said earlier. And clearly, it's not even possible to do that right now. But we need to think about, well, what's the alternatives? Do we watch our kelp beds go away? Or do we try and test a number of tools and figure out which ones work best? And obviously the scientists are, frankly, this is an advertisement, obviously. I'm in the business of trying to do things that might change things. So
just to follow up, you said right here that divers are removing thousands of urchins. I know in other parts of California they have big urchin removal festivals. And I don't know, obviously you need to be safe and maybe you have trained divers, but can the public participate in those?
Well, yes, the public can participate in those, and certainly down in the Fort Bragg area they are. And that is making a difference, in some ways in small places, and Casper Cove, for example, and that can make a real difference, and I encourage that. And I think it's important though to stay calm and to stay patient too, because we know that the disappearance of kelp is not everywhere. And so, for example, I'll give you a little tidbit of some other talk. We have been studying the mouths of rivers in our region and found that right at the mouths of rivers, we see good remnant populations of kelp. And it turns out that that fresh water flowing out in the rivers is probably knocking back the feeding urchins and, you know, kind of giving natural refuges to our kelp beds. We want to study that next, so we dig that into that further next. But that's our results so far. So yeah, I hope I've answered your question. Yeah.
I heard that there was a sunflower Seastar sighting network. Do you want to plug that again?
Sure. I don't have a slide, unfortunately, but I welcome anyone and everyone to let us know if you spot sunflower stars, and there is a network, and you can post your answers. What's that?
Through Reef Chat.
Okay, through Reef Chat. Excellent.
Are there other species that are affected by the kelp, the decrease in the kelp beds? Would you mention any?
You know, David mentioned briefly, but I'll dive in a little bit deeper here, so to speak. Kelp beds are enormously productive and diverse places with all kinds of animals. Brittle stars down in the holdfast, bryozoans on the kelp fronds, gastropods or snails feeding on them. So the collapse of many species, including important commercial species like the abalone and rockfishes, down to a barren, you know, bare rock with urchins, is catastrophic to many regions, especially in Fort Bragg. So that's one of the motivating things. And I'm actually working with a big team of scientists that are much broader than the three of us, believe it or not, where there are social scientists and there are economic scientists all coming together to work and model the system down in Fort Bragg. So we can sort of model different potential restoration plans and see what can or might work in the future.
And then we are just about out of time, but if we want to hit one more question.
I'm just wondering what happens when the kelp was gone and there's a ton of sea urchins left. Why aren't they then crashing because they're out of food?
It's a great question. Sea urchins seem to be able to, like many invertebrates, starve for a good long period of time and be okay. And so that's the bad news. The other bad news is that sunflower stars, well, and some of the other predators like otters would rather eat the urchins that have full gonads and other things inside of them. And of course, the fishermen would rather collect the big red urchins that have gonads, which they sell to the Japanese. So there's a problem there, which is unfortunately long term. And basically, scientists call these stable states, where you could have a stable state with no kelp for quite some time, because any kelp that tries to settle and grow will be immediately grazed, right, by the urchins. So we have a major problem and not an easy problem to solve. And that's why I mentioned some of the refuges, which might be starting places perhaps, for, you know, for further restoration work after those sites.
I'll just add to that, some innovative humans have gone out and on some of these sea urchin barrens and collected commercial amounts of purple sea urchins, but before they can sell them overseas, you actually have to feed them because they're so barren inside. They're still alive, so they're just able to really sustain the population on very little.
And I want to add also, as part of my mariculture class, there was a group of students, and they tried to feed them different diets, and just with the spinach, that was a good diet for them to develop the gonads or the uni that people want to buy.
Yeah, so a lot of solutions I think are needed, and I think, you know, I personally think we don't know what the future is going to hold, as David mentioned, but I'm still hopeful myself, and I am hopeful because there's a lot of people doing a lot of different things. So that's the message I would give.
Well, thank you all so much. And I just want to give a quick plug too. Gordon had the question about what can folks do. Our community just broadly, is working on like a climate action plan. Our mayor's here tonight, our council in Eureka and in the county encourages people to get involved in the process of working together, and how can we as a community reduce our impact on climate change. So yeah, come get involved in that kind of policy change as well.
Every little bit counts.
Yeah. So thanks everyone for coming tonight. Our next lecture is going to be January 21st. It is entitled Sharing Discoveries, Exploring Tropical Mammals and Amphibians Together. So we hope to see you there. Thank you.